So after the primary structure, the next level of the structure for proteins is called as the secondary structure. So in the last class, I only gave a slight description and then I stopped. So let's continue this topic. Secondary structure is of three types basically. In books you will find, like in 12 books, you will find basically two structures are mentioned, alpha and beta. So what exactly is secondary structure? We all know this long chain of amino acids, which is called as polypeptide, cannot remain like a long straight thread. Obviously it will start bending. So wherever bendings will start, there in those places of bending it is possible various type of other chemical bonds may start developing, and we call those bonds as hydrogen bonds. Now what exactly will happen? If the chain starts coiling around itself, it is possible it may develop bonds. Which bonds? Hydrogen bonds within the same chain. Intra means within the same, inter means between two. And it is even possible because in the last page we have learned that proteins can be monomeric, proteins can be oligomeric. So if it is a monomeric protein, it is not possible to have bonds between two chains because it only carries one chain. So in that case, secondary structure is established by bond formation within the same chain. So only one chain will start coiling like this in right-handed manner or clockwise manner, and finally hydrogen bonds will develop. What happens in the case of oligomeric proteins? In the case of oligomeric proteins, the bonds would be formed between two different chains. Like in case there are two chains, three chains, four chains. So these chains will start developing hydrogen bond, and in that case the bonds are called as interpolypeptide hydrogen bonds. So remember these two things. Now what are the types of the secondary structure? So the first one is called as the alpha helical structure. Now it is called alpha because it was the first one to be discovered by the scientist who discovered it. And the second one discovered is the pleated form. So he called this one as alpha because first to be discovered. He called it as beta because it was the second one to be discovered. So the alpha helical structure develops in those chains, those proteins which are monomeric. That means they carry only one chain. So definitely which bonds will develop now? Intra polypeptide hydrogen bonds. Now the bonds always develop between first and fourth amino acid. So you can understand the situation that there is a long chain, and in that chain every first amino acid is going to develop intra polypeptide hydrogen bonds with the fourth one. So first will develop with fourth, and it will continue like this throughout the chain, and this will cause involvement of like how exactly it happens. So it's very easy to understand how this bond formation takes place is that the amino group, what is amino group? This is called amino group and this is called carbonyl group. So the amino group of one amino acid is going to develop hydrogen bonds with the carbonyl group of the fourth amino acid. So I hope you are getting this point, that this is how every amino, amino group and every carbonyl group is involved in bond formation. So this will provide extra stability to which structure? Primary structure. So primary structure is stabilized, is formed by peptide bond, and then in the secondary structure this whole thing is stabilized by the hydrogen bonds. So alpha helical structure will develop turns, right hand side turns or clockwise turns, and those turns will have, each turn will have 3.6 residues or amino acid. You can say almost close to 4 amino acids in one turn will be there. The next thing is, please remember I have mentioned in the notes here, amino acid first, amino acid fourth. So they like if it is going like a chain in this manner, in the right hand side manner, clockwise manner. So first amino acid, second amino acid, third, fourth, fifth, sixth, it continues like this. So the first one will form a bond with the fourth, second will form with the next one, then third will form with the next one. So all of them are participating in the bond formation. Remember the gap between first and fourth, it means there will be 2 amino acids which will be skipped for developing the intra polypeptide hydrogen bonds. Now these bonds stabilizes it. Now next thing, there are some amino acids which cannot develop these intra polypeptide hydrogen bonds, and those amino acids are glycine and proline. Even it is possible hydroxyproline, because I have told you in the last class Hydroxyproline is a very common derivative of proline. So these amino acids are not able to develop hydrogen bonds. That's why they will cause loosening of the chain. So secondary structure will be broken by the presence of glycine and proline. So in a chain of amino acids, if there is 5 times glycine or proline present, then the secondary structure will be broken 5 times by these amino acids, because they are called as the helix breaker. Reason, they don't participate in the bond formation, hydrogen bond formation. So in those area, this coiling is going to be a irregular or random type of coil. So these bonds will cause regular or proper type of coiling. But wherever these helix breaker will come, the helix is going to break. Reason is these bonds are not possible. So these amino acids will cause random coiling in those areas where they are present. Now the second type is called as beta pleated type of the bond. Beta pleated type of the structure. Now this structure is possible in a chain which may be made up of more than one chain. That means oligomeric. So if a protein is oligomeric in type, it may have 2 chains like insulin. It may have 4 chains like in the case of we learned hemoglobin. So such type of proteins where more than 1 chains are present will develop beta pleated structure, and in that structure it is possible that the same chain may start bending like this, or it is possible 2 chains may face each other in a parallel manner. You can see N terminal, C terminal, C terminal, or the chains may be anti-parallel, N terminal this side, N terminal this side. So it is possible that these protomeres are those polypeptide chains, may face parallel, may face anti-parallel, and will develop hydrogen bonds between the chains like this. Again, same thing, these bonds will be between the amino group of one amino acid and the carbonyl group of the another amino acid. So this all is for providing stability. Now scientists discovered another type of secondary structure, and that is found in the collagen helix.